I think much has been said about the profound impact of the DNA double helix. Here, I just want to echo what Tom and others said this morning, that the simplicity, the elegance, and the beauty of this DNA double helix model is just really breathtaking, and few scientific discoveries have been featured so prominently, even in the city landscape, like shown here, in the towers and the bridges. So I talk to my students. I say, no matter how smart you are, how much you have done, in front of the double helix, we all need to be humble. Okay, so after hearing all the wonderful things about the double helix, the DNA, I want to sort of spoil the party a little bit and talk about the dark side of the DNA. I trust most of you work on the bright side of DNA, and that is how DNA replicates, how it's transcribed, how it's repaired. But DNA also has a dark side, and that is it can stimulate immune responses, and in some cases, it can trigger autoimmune responses, leading to a variety of autoimmune and inflammatory diseases. So here are the major players in my talk. This is the DNA sensing enzyme, cGAS, which is activated by binding to double-stranded DNA. The activated enzyme then converts GTP and ATP into this cyclic dinucleotide, cyclic GMP, AMP, we call it cGAM for short. This small molecule then functions as a second messenger that binds and activates this ER membrane protein, STING. STING then triggers a signaling cascade, leading to an innate immune response. So I'm going to step back and ask, why do you want to study immune sensing of DNA? And that is because recognition of foreign DNA is one of the most fundamental mechanisms of host defense. And even bacteria can do that. The classical examples are restriction enzymes. They cut bacteriophage DNA or plasmid DNA. And the more recent example, of course, is the CRISPR system, which is the bacterial adaptive immune system that cuts foreign DNA. In humans, DNA was known to stimulate immune responses more than 100 years ago. So when Mechnikov received his Nobel Prize for the discovery of phagocytosis, he noted in his Nobel lecture in 1908 that there were surgeons in France and Germany who introduced into the abdominal cavity or under the skin of the patients either warm blood serum or nucleic acid with the object of bringing to the scene a protective army of phagocytes to ward the microbes off. So DNA was known to stimulate immune responses 40 years before it was known to be the genetic material. But we didn't know how DNA stimulates immune responses until quite recently. So why do we want to have an immune system that detects DNA? First of all, it's quite versatile, with the exception of RNA viruses, which are recognized by the RIG-I pathway that I will mention in just a minute. All microorganisms, including DNA viruses, retroviruses, bacteria, fungi, and parasites, they all have DNA or require DNA in their life cycle. And this provides an almost universal mechanism to detect microbial infections. And secondly, the recognition of DNA or RNA of these microorganisms allow the immune system to detect those microorganisms that live and replicate inside our cells. We all know that toll-like receptors are very important in innate immunity, but toll-like receptors are membrane proteins with a ligand binding domain facing outside the cells, Some toll-like receptors are localized on the endosomal membrane with a ligand binding domain facing the lumen of the endosome, which is equivalent to outside the cells. So toll-like receptors, by and large, recognize pathogens outside our cells, but they are blind to those bugs that have successfully invaded our cells and replicated inside our cells. So we need another mechanism to detect these bugs that have entered our cells and replicated inside our cells. And the recognition of the DNA or RNA provides a powerful mechanism. But this mechanism has its liability because our cells have lots of DNA, and one solution that we have is to keep our DNA out of the cytoplasm. We keep our DNA in the nucleus, in the mitochondria, and in the nucleus, we wrap our DNA around histones to form nucleosomes. But this mechanism is not perfect, and when the system fails, it can lead to a variety of autoimmune and inflammatory diseases, and this is also highly relevant in cancer. So before I talk about the DNA sensing pathway, here I want to briefly introduce the RNA sensing pathway, which my lab has also been working on. So when an RNA virus, such as influenza virus or Ebola virus or SARS-CoV-2 infect our cells, the viral RNA is detected by this RNA sensing protein, RIG-I, or its homolog, MDA5. And RIG-I then interacts with this mitochondrial protein that we discovered many years ago, which we call MAPS. It stands for mitochondrial antiviral signaling. And we call it MAPS also because we are in Dallas, and Sheldon knows in Dallas, we have this basketball team called Dallas Mavericks. And at the time we discovered MAPS, Dallas Mavericks was in the NBA final. That's where the name came from. And so MAPS sits on the mitochondrial outer membrane and activates these kinases in the cytosol, IKK and TPK1. IKK activates NF-κB, TPK1 activates IL-3, and then these transcription factors move into the nucleus and function together to induce type 1 interferons and other cytokines. So that's how we fight RNA virus infection. And as I just mentioned, DNA in the cytosol can also induce type 1 interferons. But the sensor of this DNA was not known for a long time. And downstream of this DNA sensor was this ER membrane protein called STING, which was discovered by Glenn Barber in Miami and Hongbin Su in Wuhan here. And so STING can also activate these kinases to induce type 1 interferons. So a long-standing question is, what is this DNA sensor? And several years ago, we took a biochemical approach to identify this DNA sensor. And so we discovered this DNA sensing, this protein which we name cGAS or cyclic GMP-AMP synthase. And this enzyme, as I mentioned, is activated by DNA and then converts GTP and ATP into this cyclic dinucleotide, cyclic GMP-AMP. And here I don't have time to describe how we discover cGAS, but for those of you who are interested, you may look up our paper. Basically, we use very old-fashioned biochemistry to identify this DNA sensing enzyme, cGAS, and its product, cGAM. So now I will use this cartoon to illustrate the cGAS-STING pathway. So here is a cell that is infected by a virus. The virus, in this case, a DNA virus, delivers DNA into the cells. This DNA in the cytoplasm is a danger signal that is recognized by the DNA sensing enzyme cGAS. cGAS binds to DNA and becomes activated. And we and others have solved the structure of cGAS in its apo form, and it's also in its DNA bound form. So DNA binds to cGAS, and it pushes this pink activation loop, and this induces a conformational change in the active site of the enzyme. And this DNA binding also induces the dimerization of the enzyme, which is important for its activation. So now cGAS is activated, and then it converts GTP and ATP into this cyclic dinucleotide, cyclic GMP-AMP. So this is GMP, this is AMP, and has two phosphodiester bonds. One is 2'5' link, the other one is 3'5' link. So we call it 2'3' cGAP. So this small molecule then functions as a very strong second messenger that stimulates innate immune response. So now, each molecule of the enzyme produces multiple molecules of the second messenger, and each second messenger can bind to a STING dimer that sits on the ER membrane. So STING then is activated, and it will activate these transcription factors IL-3 and NF-κB, which move into the nucleus to turn on hundreds of genes that are important in immune and inflammatory responses. So that's the pathway, and here's another view of the pathway. So cGAS is activated by this B-form double-stranded DNA, and the active enzyme produces this cGAP molecule that functions as a second messenger that binds to STING. STING is then activated, and that then activates the kinase IKK and TPK1, leading to the production of type I interferons. So here I want to use this slide to highlight one important point, and that is cGAS is activated by any double-stranded DNA independent of the DNA sequence. And the crystal structure of the cGAS DNA complex shows that cGAS binds to the sugar phosphate backbone of the DNA. It does not have any direct contact with the base pair. And that explains why this is DNA sequence independent. And this property allows this single protein to detect a large variety of pathogens as long as these pathogens contain DNA or require DNA in their life cycle. We and others have shown that indeed cGAS is a sensor for DNA viruses such as herpes virus, retroviruses such as HIV, bacteria such as MTB, parasites such as malaria. And another important source of DNA is our own DNA. As I mentioned, our own DNA is usually kept in the nucleus, in the mitochondria, but under some pathological conditions, our own DNA can get into the cytoplasm, and in this case, it can activate cGAS to trigger a variety of autoimmune and inflammatory diseases. And one example of autoimmune disease is called Aicardi-Goutières Syndrome, or AGS, and these are the children with the disease. They have severe calcification in the brain, and this is because these kids produce a lot of type I interferons that damage their brain when they are born, and that's in the absence of any apparent infections. And these are monogenic diseases. The diseases are caused by mutations of single genes, and many of these genes have been identified by Yannick Krug and others. And most of these gene mutations lead to the activation of the cGAS-STING pathway. So for example, about 25% of these patients have mutations in a gene called TRAX1, which encodes an enzyme that degrades DNA in the cytoplasm. And a loss of function mutation of TRAX1 gene cripples the activity of this enzyme such that cytoplasmic DNA accumulates, and that leads to type I interferon production. So the question is whether these kids have the disease because they have hyperactivation of the cGAS pathway. And to test this, we use TRAX1 deficient mice, and these mice die within a few weeks or months after birth because they have very severe inflammation in many different organs. And we asked, if we remove cGAS from TRAX1 deficient mice, can we rescue the disease? And so here, we generated TRAX1 cGAS double knockout mice, and now 100% of the mice survive, and they didn't have any symptoms of disease. And interestingly, even if we just remove one allele of cGAS, so we reduce the enzyme concentration by about 50%, we can largely rescue the survival. And we think that this so-called haploinsufficiency of cGAS is due to a phenomenon called liquid-liquid phase separation. And so Mingjian Du in my lab showed that cGAS has multiple DNA binding domains, and they can bind to DNA through this multivariant interaction. And the multivariant interaction between DNA and cGAS drive this liquid phase separation that leads to the formation of these liquid droplets in which cGAS enzyme is concentrated and activated. And this phase separation of cGAS renders the cGAS enzyme activity highly sensitive to the concentration of both cytoplasmic DNA and cGAS. So here in TREX1 deficient cells, the cytoplasmic DNA concentration is elevated, but it's still quite low. And then this phase separation then is highly dependent on the concentration of cGAS. So if we reduce the cGAS concentration by 50%, it's then below the phase separation boundary, and the enzyme will not be activated. And that, we think, can explain this haploinsufficiency of cGAS. And so we think these results are very exciting because it basically provides the genetic proof of concept to target cGAS. So if one can develop an inhibitor of cGAS, then even if we can just inhibit the enzyme by 50%, it may be able to provide therapeutic benefits to patients with autoimmune diseases such as AGS or lupus. And work in the past 10 years or so by scientists all over the lab has shown that the cGAS-STING pathway plays a central role in many physiological and pathological processes. Yesterday, I did a PubMed search with just cGAS and STING as keyword, and it returned more than 2,000 papers. And just this year alone, there were 584 papers. And that means that every day there are 2 papers, which is very difficult to catch up. So here I want to spare you from reading all these papers and just summarize the function of the cGAS pathway with this slide. So this pathway is clearly very important for antimicrobial defense. And as I just mentioned, aberrant activation of this pathway can lead to a variety of autoimmune and inflammatory diseases, such as AGS. There's this STING disease called SAVI, lupus, and also severe COVID. Activation of cGAS by mitochondrial DNA has been shown to lead to neurodegeneration, causing Parkinson's disease, ALS, and Alzheimer's disease. We and others have shown that the activation of this pathway can lead to a non-canonical form of autophagy. And several labs, including us, have shown that this pathway, activation of this pathway can lead to cellular senescence, and this pathway is also very important for anti-tumor immunity. So now many labs and many companies are actively targeting this pathway, either by developing agonists of this pathway for cancer immunotherapy or antagonists of this pathway for autoimmune and inflammatory diseases. And I believe that in the next few years, there will be drugs coming out of this pathway that benefit millions of people. So that's the introduction. And now I'm going to sort of switch gear and turn the clock back by a billion years and talk about cGAS evolution, the evolution of the cGAS pathway, and specifically cGAS in bacterial antiphage defense. So over billions of years of evolution, there has been constant arms race between bacteria and bacteriophages, and they give rise to the enormous abundance and diversity of both. It's estimated there are 10 to the 31st bacteriophages on the planet, and that is more than all other living organisms combined, including bacteria. And as Tom and many others mentioned this morning, research in bacteriophages really give birth to molecular biology, and phage research has also led to many reagents in molecular biology like T4 DNA ligase, T7 RNA polymerase restriction enzymes, and then more recently, the CRISPR system. And so that, especially the discovery of CRISPR gene editing has led to renaissance of research in bacteriophages and bacteria. Like animal viruses, phages have DNA or RNA genomes with a double-stranded DNA genomes comprising about 95% of all phages. And bacteria have powerful immune system against phage infection, and as I mentioned, they have restriction enzymes, they have a CRISPR system. And one important advance in the cGAS-STING field in recent years is the discovery of a very large family of cGAS-like enzymes in bacteria. So Philip Kranjus at Harvard Medical School has identified about 6,000 bacterial cGAS-like proteins. And these bacterial cGAS-like enzymes can make all sorts of cyclic dinucleotides and cyclic oligonucleotides. And it was shown by Philip and Rotem Sorek in Israel that the major function of the bacterial cGAS-like enzymes is for anti-phage immune defense. So for example, in this paper from Rotem Sorek's lab, they show that this bacterial cGAS is somehow activated by phage infection. And this enzyme then converts GTP and ATP into this cGAM molecule, which in this case activates a bacterial phospholipase called CAPV. And CAPV then chew up the bacterial lipid membrane, and that causes the virus-infected cells to commit suicide. And that's the mechanism to limit the phage spread to protect the bacterial community. And so this cGAS system in bacteria is also called cBAS, cyclic oligonucleotide-based anti-phage signaling system. And interestingly, this cBAS system in bacteria are often organized as operons. And so the cGAS gene is quite often next to genes encoding effector proteins, such as the phospholipase. And one thing that is very interesting to me is that about 40% of this bacterial cBAS system, which is like more than 2,000 cBAS, have genes that encode enzymes with ubiquitin E1, E2 enzyme domains, as well as the JAP domain, which is the deubiquitination enzyme domain. And this is very interesting to me because in my previous life, I was working on the ubiquitin system. So at this time, postdoc Justin Jensen joined the lab, and one of the first questions that Justin asked is whether this bacterial E1, E2 protein can form a thioester with a bacterial ubiquitin-like protein, much like mammalian ubiquitin, which can form a thioester with E1 and E2. So Justin focused on this bacterial CAP2 protein, which is an E1, E2 fusion protein. And he cloned this cGAS operon from this strain of E. coli into the laboratory strain of E. coli, which does not have the cBAS system. And he epitope tagged this CAP2 protein such that he can do a Western blot to see whether this CAP2 protein can form a thioester with a bacterial protein. And indeed, he found that this CAP2 protein forms this covalent ADA with a 50 kilodalton protein. And this is a thioester because it is sensitive to treatment with DTT or hydroxylamine. And Justin then cut out this band for mass spec, and it turns out that it's bacterial cGAS, which is also called DNCB. In other words, the bacterial cGAS behaves like ubiquitin. And so this is kind of strange. Justin tested whether the bacterial cGAS can form conjugates like ubiquitin does. And indeed, in wild-type cells that have the cBAS system, these bacterial cGAS can form these conjugates, which are dependent on the E1, E2 activity of CAP2. So if we mutated the active site cysteine of the E1 or E2, then we don't see this conjugate formation. And importantly, the formation of these conjugates depends on the C-terminal glycine of cGAS, because if we remove this C-terminal glycine or change this glycine to alanine, we abrogate the conjugate formation. And CAP3 is a deubiquitination enzyme, and mutation of the active site residue of this CAP3 led to a slight activation of these conjugates. So the bacterial cGAS indeed behaves like ubiquitin, in that it can be activated by E1, E2 and conjugated to other proteins. And so this is quite strange because the bacterial cGAS does not look like ubiquitin at all. In particular, ubiquitin has this highly conserved C-terminal tail that has these 2 glycine residues. In contrast to the mammalian ubiquitin, the bacterial cGAS don't have these conserved diglycine residues. So for those cGAS operons, they have E1, E2, and JAP domain, they have highly conserved glycine and alanine. And for those cGAS operons, they don't have E1, E2 structures, this is not conserved. Justin then mutated the glycine residue of this bacterial cGAS into other amino acids, and he found that with the exception of this glycine to alanine substitution, which can still form some conjugates, all other amino acid substitution abrogate the conjugate formation. So the bacterial cGAS can be conjugated to other proteins through this C-terminal glycine, much like ubiquitin. So now the important question is whether this ubiquitin-like conjugation by bacterial cGAS is important for anti-phage immune defense. So here, Justin looked at infection by lambda phage, and so he compared bacteria that have the cBAS system with those that have the empty vector, and the cBAS system reduced the phage titers by more than 10,000-fold, and this activity depends on the phospholipase activity of CAV as well as the cyclase activity of cGAS. It also depends on the E1, E2 activity of CAP2, as shown here. And importantly, removal of the C-terminal glycine of cGAS phenocopy the effect of the E1, E2 mutation. And this glycine to alanine substitution has a partial effect because this mutant can still form some conjugates. And similarly, we found that this cGAS conjugation is important for immune defense by T4 phage and T6 phage. The C-terminal glycine removal basically phenocopy the effect of E1, E2 mutation. And so this ubiquitin-like conjugation by cGAS is important for anti-phage immune defense. And the question is, how does it do that? And so here, Justin looked at cGAM production in E. coli in response to phage infection. So, so the red line here basically is the E. coli strain. They have the functional cBAS system, and the green line here is a cBAS system in which cGAS cannot be conjugated. And so in response to phage infection, the cells that have the wild-type cBAS can produce a lot of cGAM molecule, and the cells that cannot conjugate cGAS, basically the cGAM production is greatly reduced. So an important function of this ubiquitin-like conjugation of cGAS is to greatly enhance its ability to produce cGAM. And so the next question is, what are the targets of this cGAS conjugation? And Justin did mass spec and identify several bacterial proteins as well as phage proteins that can be conjugated by cGAS, but he didn't have any direct evidence as to whether any of those are physiological targets. He then took an alternative approach, which is a forward genetic screen to identify phage antagonists of cGAS conjugation. So for the wild-type T4 phage, it can infect E. coli that don't have the cBAS system. It cannot infect E. coli that have the functional cBAS system, but it can infect E. coli they have the cBAS system with defective E1, E2 activity. And he mutagenized this T4 phage and to isolate those mutants that are now defective in infecting those cBAS mutants. And presumably, some of these mutants have mutations in the viral antagonists of cGAS. And this screen led to the identification of an uncharacterized gene called VS4, which is about 10 kilodalton protein. And Justin found that this VS4 protein can bind to cGAM with a KD of about 30 nanomolar. And he went on to solve the crystal structure of VS4 bound to cGAM. So VS4 forms a hexamer that binds to 3 molecules of cGAM. So each cGAM binding site is composed of 2 VS4 monomers. And these are the key residues that are involved in cGAM binding or in the oligomerization. And he validated this model by mutating these residues and tested these mutants in the in vitro assay to look at CAV activity. So CAV is a phospholipase that is activated by cGAM, and the wild-type viral protein, VS4, can inhibit CAV activation by cGAM. But if he mutated these residues that are involved in cGAM binding or involved in oligomerization, then they lost the inhibitory activity. And then Justin did bioinformatic analysis to identify VS4-like proteins in other phages, and he identified about 200 VS4-like proteins in all bacteriophage families. And importantly, the residues that are involved in cGAM bindings are conserved in these VS4-like proteins. So based on these and other results, we proposed the following model. So in response to phage infection, cGAS is somehow activated through an unknown mechanism. And the activated enzyme converts GTP and ATP into cGAM molecules, which then bind and activate this phospholipase CAV. CAV then chew up the bacterial membrane to kill the virus-infected cells, and that protect the bacterial community. However, some phages can fight back by producing viral antagonists such as VS4, and VS4 in this case functions like a sponge that soak up these cGAM molecules, and that prevents CAV activation. So in this case, the virus wins. But some bacteria would try to get an upper hand by conjugating cGAS to this ubiquitin-like mechanism. So this ubiquitin-like conjugation of cGAS greatly enhances enzymatic activity, which then produce overwhelming amount of cGAM molecules that overwhelm this viral sponge, and that this cGAM molecule then activate CAV to kill the virus-infected cells. So this basically is like a classical arms race between bacterial and bacteriophages. Basically, it's like rock, paper, scissors types of arms race between bacterial and phages. And we think that this ubiquitin-like conjugation might have arisen as a result of this arms race between bacterial and phages. And I should point out that this ubiquitin-like conjugation by bacterial cGAS was also independently discovered by another group in Colorado, Aaron Weddy's group. And this VS4 bacteriophage protein was also independently discovered by a group at UCSF led by Joe Bondi-Dinomi. And so the bacterial immunity is no less competitive than human immunity. And so this is still work in progress. It raises a lot more questions than it has answered. So key questions include, how does cGAS detect phage infection? Unlike human cGAS, which is activated by binding to double-stranded DNA, there's no evidence that bacterial cGAS can be activated by DNA alone. What are the targets of cGAS conjugation? We show that cGAS can be conjugated to bacterial and phage proteins, but it's also possible that cGAS can be conjugated to lipids, much like autophagy. How does cGAS conjugation activate this anti-phage immunity? And could there be other targets of these ubiquitin-like conjugation enzymes in bacteria? So we still have a lot of work to do. Okay, so in closing, I just want to highlight the fact that cGAS now join restriction enzymes in CRISPR as an important anti-phage immune defense system in bacteria. You can think of CRISPR as the bacterial adaptive immune system, and cGAS as one of the innate immune system in bacteria. And interestingly, in humans, we no longer rely on restriction enzymes or CRISPR to defend us against virus infection, but we still rely on the cGAS system to protect us from virus infection. So this pathway has been conserved for billions of years from bacterial to human. So this is a human cGAS here. This is Drosophila cGAS. This is a C. anemone cGAS. There's a unicellular organisms. They also have cGAS. Archaea have cGAS. And of course, bacteria have a lot of cGAS. And cGAS apparently originated in bacteria, and the antiviral function of cGAS has been conserved for billions of years. And so I think I have acknowledged some people who did the work, and in particular, the new work on bacterial cGAS was done by Justin Jensen with help from To Li and Feng Du. And thank you very much.